Hello everybody, I am Dr. D. Kavita, Assistant Professor, Department of Biochemistry, Biotechnology and Bioinformatics of National Institute for Home Science and Higher Education for Women, Coimbatore. Now you have chosen to study the module on the design of bioreactors, medium composition and microorganisms used in food industry. This module describes the design of bioreactors, the composition of media for fermentation and the microorganisms used in food industry. Design of bioreactors requires integration of microbiology, biochemistry, process engineering and economic analysis. Media formulation is an essential stage in the design of successful laboratory experiments, pilot scale development and manufacturing process. After completing the module, you will be able to design the type of bioreactors suitable for the fermentation process. You will be able to define the media formulation for any fermentation processes. You will understand the types of microorganisms used in food industry. Let us see about bioreactors. Bioreactor design is an integral part of bioprocess engineering. Bioreactors are controlled environments for microbiological and biochemical reactions to produce value-added products or to treat waste streams. The aim of bioreactor design is to produce product with specific quality attributes at minimum cost. There are many important points that have to be considered while designing and constructing a fermenter. The fermenter should be reliable in long-term operation with aseptic conditions. There should be adequate aeration and agitation for the microbial metabolism. The bioreactor should be designed to use minimal labor, low power consumption suitable for wide range of processes and should have smooth internal surfaces. Various systems of control for pH, temperature, pressure, etc. should be provided. To facilitate scale-up processes, the geometry of the smaller and larger vessels in the pilot plant and large-scale plant should be similar. This diagram shows the various components of a bioreactor. The materials used in construction of a fermenter are glass and steel, which has to withstand repeated steam sterilization cycles. In a small scale, glass is used because it is non-toxic, corrosion-proof and transparent. Also, it gives smooth surfaces. Pilot scale and large scale industries use vessels made up of stainless steel. The corrosion resistance of stainless steel is due to the existence of a thin hydrous oxide film on the surface of the metal. Now, let us discuss the types and classification of bioreactors. Bioreactors are generally classified into two broad groups: suspended growth bioreactors and biofilm bioreactors. First, let us see about the suspended growth bioreactors. The reactors use microbial metabolism under aerobic, anaerobic or sequential anaerobic or aerobic conditions to biosorb organic compounds and biodegrade them to harmless residuals. The microbial activity in the systems produces biomass that is removed by gravity sedimentation with a portion of the settled biomass recycled to maintain a desired mixed liquid suspended solids concentration in the bioreactor. Example: batch reactors, CSTRs, that is continuous stir tank reactors, plug flow reactors. This diagram shows the functioning of a suspended growth bioreactors. The second type is the biofilm bioreactors. In biofilm reactors, most of the microorganisms are attached to a surface and in this manner kept within the reactor. Biofilm is also used regularly for wastewater treatment and the bacteria can either absorb or break down toxic substances in the water. The different kinds of biofilm reactors include membrane, fluidized bed, packed bed, airlift and upflow anaerobic/blanket reactors. This diagram shows the various biofilm bioreactors. Having discussed about the bioreactors, now let us look into the importance of media formulation. The fundamental knowledge available about the microorganism, substrates and process represent the basis on which a rational approach for the design and formulation of media for microbial processes can be attempted. In this respect, it is necessary to analyze critically the main objective to be optimized, that is yield, productivity, quality of final product, etc. The first step of medium development is concerned with the decision to be taken about the adequate components to be used, followed by the calculation of their concentrations. All microorganisms require water, sources of energy, carbon, nitrogen, mineral elements and vitamins plus oxygen if aerobic. On a small scale, it is easy to devise a medium containing pure compounds. But on a large scale, one must use sources of nutrients to create a medium which will meet as many as possible of the following criteria. The media will produce the maximum yield of product or biomass per gram of substrate used. The media will produce the maximum concentration of product or biomass and allows the maximum rate of product formation. The yield of undesired products will be the minimum. It will be of consistent quality and be readily available throughout the year. It will cause minimal problems during media making and sterilization. Cane molasses, beet molasses, cereal grains, starch, glucose, sucrose and lactose can be used as carbon sources, and nitrates, constip liquid and soybean meal can be used as nitrogen sources, as all of these meet most of the above criteria. The medium selected will affect the design of the fermenter to be used. The microbial oxidation of hydrocarbons is a highly aerobic and exothermic process. Thus, the production in fermenter had to have a very high oxygen transfer capacity coupled with excellent cooling facilities, which can be solved by developing an airlift fermenter. The composition of the medium also plays an important role in fermentation. A medium with a high viscosity will also need a higher power input for effective stirring. The medium may also influence pH variation, foam formation, the oxidation reduction potential and the morphological form of the organism. It is also necessary to provide the precursors or metabolic inhibitors. The medium will also affect product recovery and effluent treatment. Undefined complex natural materials have been used in fermentation process because they are much cheaper than pure substrates. Difficulty in product recovery and effluent treatment occurs as all the components of a complex nutrient source will not be consumed by the organism. The residual components may interfere with the recovery and contribute to BOD of the effluent. So, we use defined media components because although they are more expensive, pure substrate give more predictable yields from batch to batch recovery. Purification and effluent treatment are also much simpler and therefore cheaper. Process improvements are also easier to detect when pure substrates are used. The constituents of the medium must satisfy the elemental requirements for cell biomass and metabolite production, and there must be an adequate supply of energy for biosynthesis and cell maintenance. Equation based on stoichiometry for growth and product fermentation for an aerobic fermentation is carbon and energy source plus nitrogen source plus oxygen plus the other requirements will lead to biomass plus product plus byproducts plus carbon dioxide plus water plus heat. This equation should be expressed in quantitative terms, which is important in the economical design of the media if component wastage is to be minimized. Some nutrients are frequently added in substantial excess of that required. Example, phosphorus and potassium. The concentration of phosphorus is raised in many media to increase the buffering capacity. Some microorganisms cannot synthesize specific nutrients. Example, amino acids, vitamins or nucleotides. So, they should be incorporated into the medium in adequate amounts. The carbon substrate has a dual role in biosynthesis and energy generation. The carbon requirement for biomass production under aerobic conditions may be estimated from the cellular yield coefficients, which is represented as Y. Y is equal to quantity of cell dry matter produced divided by the quantity of carbon substrate utilized. The other major nutrient which will be required is oxygen, which is produced by aerating the culture. Water is the major component of all fermentation media and is needed in many of the ancillary services such as heating, cooling, cleaning and rinsing. It should be pure and pH, dissolved salts and effluent contamination should be assessed while selecting the water supply. The water may be treated by deionization or other techniques and salts added or the pH adjusted to favor the desired product. The reuse or efficient use of water is normally of high priority. Energy for growth comes from either the oxidation of medium components or from light. Most industrial microorganisms are chemoorganotrophs. Therefore, the commonest source of energy will be the carbon source such as carbohydrates, lipids and proteins. Some microorganisms can use hydrocarbons or methanol as carbon source. After medium preparation and experimentation, the application of the most appropriate statistical optimization methodology will make it possible to attain the optimal medium. Medium design and formulation for plant cell processes are still carried out on an empirical basis owing to the lack of fundamental knowledge. However, some general guidelines can be given for growth and production media considering the experimental evidence available about the functions and influence of the medium components for promoting growth and product formation. Adequate manipulation of the carbon, nitrogen and phosphorus sources and phytocarbons and as well as the inclusion of precursors and elicitors in the medium represent the best strategy for improving medium development for plant cell processes. The development of cost-effective medium supplies for mammalian cell culture production of proteins is a key element for a commercially successful process. Medium development is a task that requires experience, time and resources. Now, let us see the various types of microorganisms used in food industry. The industrial processes of fermentation involves growing large numbers of microorganisms for various chemical or pharmaceutical productions. Microorganisms are usually incubated under specific conditions in large tanks called fermenters, which leads to the chemical conversion, also referred to as bioprocessing. Most commonly used microorganisms are yeast, bacteria, molds or a combination of these. A good example of microorganism usage in food production is the process of fermentation, which results in the production of organic acids, alcohols and esters. These help to either preserve the food or produce distinctive new food products. Let us discuss the bacteria involved in food production. Bacteria are used to make a wide range of food products. The most important bacteria in food manufacturing are Lactobacillus species, also referred to as lactic acid bacteria. The lactic acid bacteria ferment lactose, a form of sugar, to lactic acid. These bacteria are usually found in milk and are also used as starter cultures in the production of cultured dairy products such as yogurt. Lactic acid bacteria are divided into two groups according to their shape, lactococci and lactobacilli. In dairy industry, microorganisms used in the production of cultured dairy products such as yogurt and cheese are called starter cultures. As these microorganisms grow in milk, they convert lactose into lactic acid. During ripening of the product, the enzymes produced by the starter culture microorganisms are responsible for the final product's aroma, taste and texture. Starter culture also inhibits growth of undesirable organisms. Sometimes these starter cultures are made from just one type of microorganism, in which case they are called simple or defined cultures. When they are made of many different types of microorganisms, they are called mixed or compound cultures. Starter cultures prefer different temperatures. Lactic acid bacteria are also used in pickling food products. During cucumber pickling, lactic acid bacteria grow and digest sugars in the cucumber, producing lactic acid. This acid gives the pickles their characteristic sour taste and it controls the speed of spoilage causing microbes. By digesting the sugars, lactic acid bacteria remove a potential food source for bad bacteria. That is why pickling has been used as a food preservation method for centuries. A starter culture is essential to make cheese. As the culture grows in the milk, it converts the sugar lactose into lactic acid, which ensures the correct level of acidity and gives the cheese its moisture. As the cheese ripens, the culture gives it a balanced aroma, taste and texture. It is also responsible for the holes in cheese such as emmenthal. Choosing the right mixture of culture is essential for a high quality cheese. In yogurt and other fermented milk products, the microbial culture is responsible for the taste and texture of the final product. Depending on the acidity, the product will have either a mild or strong taste, and the viscosity depends on the quantity of polysaccharides, chains of sugar molecules that are produced. In recent years, probiotic cultures have become popular in dairy products because of their health benefits. These cultures are all very carefully selected strains, and there is good evidence that they help to improve digestion, safeguard the immune system and keep the body's intestinal flora in balance. Coming to the application in meat industry, the meat starter cultures are used to make dried fermented products such as salami, pepperoni, chorizo and dried ham. Lactic acid bacteria develop the flavor and color of the products. In addition, a wide variety of molds are used to ripen the surface of sausages, preserving the natural quality of the product and controlling the development of flavor. In wine industry, yeast are responsible for the fermentation process, which produces alcohol in wine. However, lactic bacteria also play an important role as they convert the unstable malic acid that is naturally present in wine into the stable lactic acid. This conversion gives the stability that is characteristic of high quality wines that improve on storage. Next, we shall discuss the importance of yeast in food production. Yeast have two main uses in food production: baking and making alcoholic beverages. They have been used in this way since ancient times. There is evidence that ancient Egyptians used yeast in bread making, and we have been making fermented drinks like beer and wine for millions of years. In baking, the baked goods like bread rise because of the presence of yeast as a raising or leavening agent. The most common yeast used in bread making is Saccharomyces cerevisiae. It feeds on the sugars present in the bread dough, producing the gas carbon dioxide. This forms bubbles within the dough, causing it to expand. The production of carbon dioxide by yeast is needed to make the dough rise or increase in volume. This process is called leavening. Leavening makes the dough lighter and more easily digested. Yeast cells also produce different types of organic acids and alcohol, which make bread taste better. Baking evaporates any alcohol and deactivates the yeast. It also causes bubbles of carbon dioxide to move through the dough, giving the bread a spongy texture after baking. Other ingredients in the mixture have an effect on the speed of the fermentation, that is, sugar and eggs speed it up while fats and salt slow it down. Brewing, in several different yeasts are used in brewing beer, where they ferment the sugars present in malted barley to produce alcohol. One of the most common is Saccharomyces cerevisiae, the same strain used in bread making. This is used to make a type of beer and is known as a top fermenting yeast, as it forms a foam on top of the brew. Bottom fermenting yeasts such as Saccharomyces pastorianus are more commonly used to make lagers. They ferment more of the sugars in the mixture than top fermenting yeast, giving a cleaner taste. In wine making, the alcohol is formed by the fermentation of the sugars in grape juice with carbon dioxide as a byproduct. Yeast is naturally present on grape skins, and this alone can be sufficient for the fermentation of sugars to alcohol to occur. Pure yeast culture, most often Saccharomyces cerevisiae, is usually added to ensure the fermentation is reliable. Sparkling wine is made by adding further yeast to the wine when it is bottled. The carbon dioxide formed in this second fermentation is trapped as bubble. Next to bacteria and yeast, molds have a major role in food production. Molds are essential components of several food products such as some cheeses, sausages and soy sauce. In cheese making, three main types of cheese rely on molds for their characteristic properties. They are blue cheese, soft ripened cheese and rain-washed cheese. To make blue cheese, the cheese is treated with a mold, usually Penicillium roqueforti, while it is still in the loosely pressed curd form. As the cheese matures, the mold grows, creating blue veins within it, which gives the cheese its characteristic flavor. Examples include Stilton, Roquefort and Gorgonzola. Soft ripened cheese such as brie and camembert are made by allowing Penicillium camemberti to grow on the outside of the cheese, which causes them to age from the outside in. The mold forms a soft white crust and the interior becomes runny with a strong flavor. Rind-washed cheeses like Limburger also ripen inwards, but here, as the name suggests, they are washed with brine and other ingredients like beer and wine, which contains mold. This also makes them attractive to bacteria, which add to the flavor. In meat fermentation, a wide variety of molds, that is Penicillium chrysogenum and Penicillium nalgiovense, are used to ripen the surfaces of sausages. The mold cultures play a role in aroma formation and improve the texture of the sausages. They also contribute to shortening of the ripening period and preserving the natural quality, and in that way expanding the shelf life of the meat product. Inoculations of sausages with molds were traditionally done with the indigenous flora of the slaughters, the so-called house flora. In soy sauce is made by mixing soybeans and other grains with the mold, either Aspergillus oryzae or Aspergillus sojae, and yeast. Historically, this would have been left to ferment in the sun, but nowadays it is mostly made under industrial conditions. The key flavor ingredients formed in this process are salts of the amino acid glutamic acid, notably monosodium glutamate. Thus, there is a growing demand for the production of foods from various microorganisms. This module has presented the overview of the bioreactor design. Media formulation is an essential stage in the design of successful laboratory experiments, pilot scale development and manufacturing process. This module has also explained the role of different types of microorganisms used in the food industry.